I'm going to differ a little bit from the whole perspective of model fitting. But let's see how it goes. So I'm going to talk about how the connectivity structure of the networks influences dynamics and how the models can help us change our perspective on what we measure. This work is done with my former PhD students, Sima Mengiste, Sebastian Spreitzer, who is about to defend his thesis, and master student Shreya, Leo, and Fawaz. And Andrew has been helping a lot with this. So I want to start with this quotation. I think it's important to hide behind a big name because I'm going to say something maybe which is controversial. The purpose of models is not to fit the data, but to sharpen the question. And let's see if we can do something like that. And when I think of a question, and when I think of models, to me, models are tightly coupled with measurement. In fact, it's the models that give us some way to measure things. They give us the measuring tape, but they also tell us which variables are important to measure. So I think this loop should be kept in our thinking when we are modeling, whether we are fitting the models or we are testing the models. And would be great if we can use models to improve our measurements or change our perspective on measurements. So the measurement I'm going to focus on is the connectivity measurement. And Allen Brain Institute and other labs, they have been collecting huge amount of data. There are classical results about this. And we know a lot about brain's connectivity at very large scale, interregional connectivity, for instance, where we know that it's not random. It has some structure, and that structure could be maybe small world, maybe hierarchical. We have specific anatomical structures in the brain, some brain systems like basal ganglia or cerebellum in this example. And if we zoom in further, we find that the connection probability is largely distance dependent, depends on neuron morphology, of course, as well as their density. So across layers, it might be different just because number of neurons differ. In some species, neurons, they make long-range patchy connections, as shown in this primate data. And finally, if we really zoom in, then we find that there are microcircuits that go across the layer. So this is slight improvement on the classical microcircuit that Kevin Martin and Douglas were discussing, or I think Mountcastle already was thinking. Now we have better definition of cell types. Therefore, we can talk about a slightly different version of this microcircuit. And the question is that how do these different features of connectivity, they influence the dynamics? Do they only bring in any quantitative change, or are they also capable of bringing qualitative changes? And at least in my thinking, it would make more sense to focus on measurements that bring in any qualitative changes. So how do we ask this question? We have models where this kind of data could be integrated. And those models are biological neural network models where neurons are relatively simple. Synapses are also very simple, leaky integrate fire neuron, for instance, and static synapses. And people have been looking into various types of topologies, feed-forward networks, random networks with local connectivity, as well as patchy connectivity. And we have also seen models with just inhibitory neurons, inhibitory excitatory neurons, but without excitatory to excitatory connections, as well as the classical neocortex model where there are all four connections allowed. The question is that now if we put in structure, will something happen? And will something happen to the network activity? And when we think of the network activity, we can define a variety of descriptors of this. There are classic ones like firing rate, synchrony, spike time regularity, oscillations. But more recently, we have data as well as one can measure these, the temporal sequences, low dimensional representation that Carson was talking about. So all these things could be measured. And then the question would be, what kind of structural features are those that influence any one of these? And they influence them in some qualitative manner. So I want to start with something that we all very well understand, that is the random network without any structure. And for this, there is this very classic result that excitation inhibition balance is the key determinant of network activity. We can change the balance of excitation inhibition, and we can obtain different kind of activities, starting from almost Poissonian state that is asynchronous irregular to a clock-like behavior, synchronous regular. In between, depending on what value you choose, you can get something called synchronous irregular and maybe oscillations as well. So by changing excitation and inhibition, you can get variety of states, but they will fall in one of these categories. And the key determinant is balance, not the connectivity, because to a great extent, the effect of shared connectivity could be canceled by the balance of excitation and inhibition. So how does this idea translate to something more complex as we go away from Ado-Shreni type of networks to something like small world or scale-free topologies? Will there be any big difference? Now here it might seem that we have put distance-dependent connectivity in small world, but that's not the idea, it's just the representation. When you are analyzing this, you can scramble it, and main focus is on the connectivity degree distributions. So first question we ask is that, is it important to worry about whether the topology is random or small world or scale free? And to do that, we of course wanted to simulate these dynamics, and it was very easy to tune them into similar kind of activity states, such that if you just looked at these activities, it would be difficult to say whether it comes from a small world or scale free or Ado-Shreni type of network. So that's definitely not a way to distinguish it. So we took a somewhat curious approach to test this idea whether topology matters or not. And what we did was we said that, okay, let's start to degenerate these networks, because after all, we are interested in the structure of the network. So we start with an average connectivity of 10% and systematically prune this network and go down up to 1%. Since this network is already tuned to operate in inhibition dominated regime, we notice that the firing rate increases as you degenerate or remove synaptic connections. There are 4 columns here. They tell us, or these 5 columns, they refer to 5 different strategies we use to remove connections in the network. And this was for Ado-Shreni network, and we knew that this is what will happen. But if you look into small world or scale-free models, they're also exactly the same thing happen. Numerical values differ, but the way the activity changes, meaning it goes from low to high activity as we degenerate it, that's pretty much the same for a small world scale-free. And to our great surprise, this was not only the case for synaptic loss, it was also true for neuron loss. So we also removed neurons in different ways. In almost all cases, we had basically the same behavior, that activity, whatever is the normalized value in the baseline network, it only increases. There are small differences here for these 2 strategies, but these 2 are very specific because we are removing neurons which have high degree or low degree. And that doesn't really influence the network in a big way. So therefore, these 2 perturbations perhaps were not very clever choices. So this is about firing rate. And if we go to some other higher order properties like spike irregularity or synchronization of spikes, there also we have qualitatively the same behavior irrespective of the network we choose. So for instance, neurons, they start with irregular firing and they tend to become more regular as we remove the connections because they are mainly now externally driven. Synchrony, it starts with somewhat low synchrony, and I mean, it was already tuned to be in an asynchronous state, but from that point on, it becomes a little bit less synchronous because again, it is largely driven by external input once connections have been removed. So this was all expected. But it was not expected for small world and scale-free topologies, and especially not for both synaptic and neuronal losses. So this tells us, at least up to this point, that it may be that the topology is not important, even though it's such a big descriptor of the network structure. But it turns out when we look at the dynamics and dynamics of particularly spiking neurons, then it seemed to not matter. Then question was that what does matter? So we had about 10,000 different networks. So we said, okay, fine, let's measure for each of these networks some structural measures, like shared connectivity, average degree, variance of that, spectral radius, number of neurons, and the connection density. And we also look into dynamics measures, for instance, average firing rate, the variance, population synchrony, as well as pairwise synchrony and irregularity. When we do the regression on between these set of variables, we find that the best descriptor is what is called effective synaptic weight. And what is effective synaptic weight? Well, that's essentially the sum of excitatory input to a neuron and the inhibitory input to the neuron. So essentially, ESW is nothing but the EI balance. And this correlates beautifully, almost linear in these this parameter range, that firing rate will increase, asynchrony will decrease, and irregularity will decrease as well. So it turns out that in random networks, when we don't have this notion of space, doesn't matter what topology we start with, the main descriptor is the EI balance and not anything else. There are other contributions, for instance, from the variance of the EI balance, somewhat from the shared inputs, but nothing really covers as much as the ESW does. Then we were wondering, like, what happened? Topology is so important. So how does the network change if we remove connections the way we were doing it? So then SEMA designed these different descriptors of randomness, small worldness, scale freeness, and checked whether the networks, to what extent they were random, to what extent they were small world or scale free. And it turns out that in most cases, whichever perturbation we bring in, so wherever it is red, it means that the network retains its character. Wherever it's blue, that means the network has changed its character. So in most cases, you notice that the network is no longer the characteristic network that we built. And now in retrospect, it sounds quite obvious because all these 5 different strategies that we use to remove connections, they are not obeying the small world network algorithm or scale-free network building algorithm. So we are actually destroying that particular rule, and therefore topology by itself becomes a very, very fragile concept. So at least it looks like from this work, to whatever extent these models are biologically plausible, it looks like that topology perhaps is not the interesting descriptor. What perhaps matters most is the effective excitatory and inhibitory balance. I've only said something about the magnitude of this balance. What also matters is timing of the balance, but that's for some other point. So this was about random networks without any space. What about spatial networks? We do have data about that. And if we take this rule that the connection probabilities, they change as a function of distance, and maybe there are patchiness. So you could ask, like, what kind of shapes are important? What should be this kernel? Or is this kernel any particular, or is this kernel of any particular importance? And for that, we have a very nice, let's say, theoretical framework that of neural fields that we can use to analytically get some insight into this problem. So that's basically the neural field equation, where you define the activity of a neuron at some location as a function of time, and it depends on the connectivity kernel, WXY being the shape of this kernel that we have put in the model. And that's just the firing rate. And you can put external inputs, either homogeneous or localized. And to solve this, at least for homogeneous case, when every neuron is identical, connectivity kernel is homogeneous and so on, for that we can actually do some calculations. And we can check what is the stability of the homogeneous solution. Long story short, what this tells us is that if we solve for the homogeneous solution and check for the stability, like if we perturb the homogeneous solution, what comes out, what becomes important is the Fourier spectrum of the connectivity kernel, because this is what is defining the stability of the system. And in that there are 2 parameters. One is the slope of the neuron transfer function, and other is the Fourier spectrum of the connectivity kernel. And essentially, Fourier transform of the connectivity kernel can tell us everything that we want to know about the spatial structure of the activity, whether it will be periodic, aperiodic. If it is periodic, what is the spatial frequency, and so on. And this already at this stage, we can imagine that if we want to have some spatial frequencies, our connectivity kernel should be non-monotonic. And in fact, that's what you can calculate as well, that if you start with a Gaussian shaped kernel, its spectrum doesn't have any peak at non-zero frequencies, K here being the spatial frequency. It doesn't matter how you scale it, all you get is a single bump, meaning a 0 frequency, homogeneous activity, which decays out. However, if you introduce non-monotonicity in the kernel, that is, for instance, you make it Mexican hat, it's something that we often get when we sum up excitation and inhibition. In that case, depending on the actual parameters of the shape, you can get a peak at a non-zero frequency. That means that Mexican hat shaped kernel, they are able to, or they will be able to generate spatial patterns. But the key here is that the way this Fourier transform changes, it heavily depends on how non-monotonic the connectivity kernel shape is. And this we could then demonstrate in a simulation of an inhibitory network, because in excitatory network, E and I will sum up and will always create something non-monotonic. So we checked it in an inhibitory network. If we start with Gaussian connectivity, we don't get any spatial structure. It's all homogeneous, randomly distributed activity. But if you put something like a donut-shaped connectivity, at some input, we start to see patterns. These are spatially compact clusters. And nothing really changes if we now vary the parameters of this kernel. As long as the shape is maintained, we can vary it, the shapes or the scale parameter, because we had gamma function as our kernel. And you can get these nice patterns. We can vary their frequency, but nothing qualitative happens. Once you have chosen a non-monotonic shape, that's what you will get. And that's about it. So now we can take this and we can check whether the patchy connections are somehow more important. Well, with this heuristic that we have built, that what matters is how non-monotonic the connectivity kernel is. Here, the red will be excitation, blue will be inhibition, and the purple will be then the summation of the two. So by putting a patch, what we are doing is adding an extra non-monotonicity in this. That means that depending on the distance between the main lobe and the patch, we are going to add an extra spatial frequency in this model. So that's the heuristic that Fawaz had. And then he actually checked whether it matters where you put the patch. The patch is close by or far away. Of course, if it is far away, it should be a lower spatial frequency. If it is close by, it is higher spatial frequency. But if you are really, really close, that means the patch automatically becomes part of the main lobe, then you will see nothing. And this is exactly what we could see in the Fourier spectrum of this connectivity kernel. And to check this, then we again run the simulations. And Fawaz, who is very artistic, he came up with this kind of illustration where he puts all the neurons at the center and their activity is represented as a radius in the color. So as soon as you put a patch, the stationary activity becomes unstable and you get 3 patches. But the analysis also predicts that if you move the patch location, you should do something to the number of patches that you're going to get. And if it is close by, then you should get a lot more patches because spatial frequency is higher now. So in this case, the movement of the patch is slowed down. However, we very quickly end up in a situation where there are, at least, I think in this case, it's 5 lobes that emerge, and they are all unstable. So what it tells us is that adding patches to the connectivity does 2 things. It increases the spatial frequency. Second, it makes those spatial patterns unstable. Now, of course, it is a bit tempting to think that this model could be used to explain the emergence of traveling waves in the cortex. We know that they exist in V1, V4, but what is needed is that we need to somehow spatially filter the activity to see these patterns. I'm not so sure because what I showed you was that patches are, I think, mainly a feature of a primate brain. And this data that is on your screen, at least some of this data is coming from mice. I think this V4 data coming from Carandini lab, but this one is V1 from monkeys. So I think in both cases, there are traveling waves and some kind of connection asymmetry could be one reason why we see these patches. So there's a question from Shinya Ito that's about the shape of the connectivity kernel. So right now I've shown you 2 cases, one with one dimension. Those were the movies that I showed you. The previous work with the inhibitory network, there we considered a 2D kernel. As far as I know, nobody has considered a 3D kernel in neural field equation, but I may be short of my reading here. So this was all about homogeneous networks. So we have talked about random networks without space. There it's EI balance. We have talked about spatial networks, and it turns out there all that matters is the non-monotonicity or the shape of the connectivity kernel, not so much the connection probabilities. But what about now heterogeneous networks? Because of course the brain is very, very heterogeneous. Everything is inhomogeneous. So by studying networks, we were pretty much convinced that what really is going to be important is the connectivity. So we should be focusing on the heterogeneity of the connections. So we thought that we could make this connectivity that is often considered isotropic in space. that this could be made asymmetric. After all, neurons, their dendritic or axonal arbors are not spatially isotropic. They are polarized to some extent. So what Sebastian did then is that he said that we could tilt this kernel a little bit so that a small fraction of connections are made in a direction phi, for instance. But then the question came up, what should be the phi of all the neurons? Should it be random? Should it be identical, or something in between? So then we did all 4 combinations, meaning there's no phi, symmetric connectivity, homogeneous, meaning all neurons are slightly polarized in one direction, random, where neurons have different polarizing directions, and Perlin. Perlin is a gradient noise, and this noise allows us to control the spatial domains where the preferred angle of these neurons, connectivity could be controlled. So we could control the size of these domains here. A fun fact here is that Perlin is a computer scientist who designed this noise for Disney, and he won an Oscar for this noise. So we put these different connectivity preferences in our network, and this is what comes out. So now each color dot is a neuron, and whenever the color shows up, that means the neuron is spiking. Here, there is no preference in the symmetric case. In random case, neurons are oriented in different directions. In the homogeneous case, they are oriented in one direction, and so on. So in this case, when neurons have preferred direction, which is random, you don't really see any difference. So that doesn't make any big difference. If everybody is projecting in the same direction, we get this beautiful traveling wave now. And what I like the most is this Perlin case, where we have local domains, and therefore what we get is this local sequences. So every little piece of the network that you will look at, you will see a different set of sequence. And since this is now activity generated by giving common input to all the neurons, there is dependence. But you can imagine that if inputs are slightly modified in different parts of this network, these sequences would be pretty independent of each other. So this gives a qualitatively different dynamics in the network, something that you wouldn't expect. So this suggests that what really matters is somehow this correlation in the property or connectivity of neighboring neurons. On one hand, this kind of activity, this reminds us of Turing patterns. This is exactly what happened in Turing patterns as well, where excitatory and inhibitory substances diffuse at different velocities, and they create all kinds of patterns. What we have done here is that created some correlation in their diffusion constants. And as a consequence, we get these pathways along which the activity flows. But we did not change anything in the degree. In Perlin case, there is this little clouds that appear where the degree of some neurons is high. And I'll come back to this, what it means. So we were again tempted now to relate this model that we basically cooked up, starting with the assumption that neurons are actually not isotropic in their connectivity. So we are aware that people can measure a lot of neurons simultaneously in the brain these days. And one typical analysis that is shown is that there are sequences. This is not the same as Juliana was showing, because there it was really individual spiking activity. Here they look arranged according to the rate, according to the peak rate of these neurons. And there you see some sort of sequential activity. And if we do the same analysis with our network, randomly sampling some neurons, we do get something similar. I mean, it's not identical, but it's in the same direction. And if we now silence the network and activate only a bunch of neurons, then you can get a beautiful sequence, which is really a sequence of spikes. They last for a few tenths of milliseconds, depending on the domain of the Perlin noise we choose. So we did some sort of virtual anatomy on these networks to really get into the bottom of why these sequences emerge. And what we found is that in homogeneous network, you really create these little pathways, but essentially it's a single path. However, with this gradient noise, we create many independent paths. So here what we did is that looked into a bunch of neurons, found out where they together project most likely, and then we create these chains. So in a way, you could think that this is also an algorithm to embed feed-forward structures in an otherwise random network. But this is, I think, only part of the story. We went back and again looked at the effective inputs to these neurons, something that Seema was doing by summing up total input coming into these neurons. And what Leo found is that when we look at this, then it turns out because of this local patches, what we have created is local domains with high excitation and low excitation. And if I could zoom here, you would see these arrows which are projecting in a particular direction depending on the gradient. So essentially, it turns out these sequences or these these anatomical pathways that have come up, they are actually the pathways of high excitability or low excitability. And the activity just glides along those pathways. So once again, we are back to the same thing that we started with in a random network, that eventually what really matters is excitability or EI balance of these networks. But on top of that, when we are talking about inhomogeneities, we should be considering that inhomogeneities, they should have some spatial correlation. An individual with a different property will be basically averaged out. So you need to make local groups so that you can be heard of. Up to this was just an exercise where we built a connectivity rule, and according to that, we designed these networks, and they have this nice property. But of course, if these kind of ideas, they have to be viable for the brain function, then there must be some way that this could be dynamically changed. And of course, synaptic plasticity could do it, but that's not fast enough. So one place we turned to was neuromodulators. Very recently, it has become possible to image neuromodulator release patterns. And what that tells us is that neuromodulatory release patterns, so this is in motor cortex, these are the possible receptor sites. So it turns out that there is locally patchy release or uptake of neuromodulators. So that is one observation. Second observation is that this is now in striatum, where animals were trained to do some conditioning task, instrumental conditioning or Pavlovian. And in that, when they observe the dopamine release pattern or activity of the dopaminergic axons, it's not really dopamine, so to say, turns out there are waves. And they have a preferential direction depending on the task. In Pavlovian task, it moves in one direction. In instrumental task, it moves in another direction. So neuromodulatory substances, besides changing and gating plasticity, they also influence neuron membranes. So what one can imagine happening is that even though the network may have completely isotropic structure, just because these patches of dopamine or any other neuromodulator, they are released in the spatial manner, that locally they are going to change neurons' excitability and create these local hotspots, which will be naturally spatially correlated. And these hotspots will have different EI balance compared to their broader neighborhood, and that will give rise to sequential activity as well. And depending on the change in the neuromodulatory pattern, you can easily switch the pattern structure, etc. Now, this was all about networks, still considering something very restricted in the sense that only two types of neurons, E and I. We, of course, know that that's not the case. There is a particular connectivity rule that, for instance, SST and PV and VIP neurons observe. So there are these motifs, and we know that these motifs do make a difference. There are several papers. We have done some work on this. And also there's a beautiful paper from Henrik Spreckler's group about this, explaining how these motifs, they function and qualitatively change the dynamics. But if you analyze that carefully, it turns out that this particular motif, for instance, this inhibition between two inhibitory populations, what this actually ends up doing is that changes the nature of the EI balance in the network, and therefore it shows up as a qualitative change in the dynamics. So to summarize here, what we have seen now, integrating this data into network models, that if we are thinking of qualitative changes in network dynamics, then really the key descriptor that comes out is the EI balance. And EI balance could be modified in many different ways. It could be non-monotonic connectivity kernel feature, it could be spatial asymmetry, but if you are interested in inhomogeneities, then they have to be somehow spatially correlated. So what then these models tell us about reformulating or sharpening our questions? At least from my side, I believe that it's of course very important to chart out details of the connectivity of a brain. But I think from the dynamics perspective, perhaps it's more important to know what are those connectivity features that are going to change the symmetry of EI balance. So whatever breaks the symmetry of EI balance, that's going to be most important. So that's where I would like to close it, that at least to me, when we use these models and not really fit the data, I didn't really try to fit the data to train it to obey certain or match up with certain activity. We just took this qualitative information that connectivities of this type and activities about that much and so on. If we go like that, I think we can make this iterative loop of measurement and models and try to get to the most crucial variables that we should be measuring. Thank you so much.